going to get my phone. Okay, so we'll get going again. So Zoltan went through a lot of the conceptual issues in pre-registration, and I was going to go through in a little bit of detail of how you can actually make those choices and some tips and advice for constructing your pre-registration. So Zoltan left on one of his final slides on this issue of power and sample size. So if you are determining your sample size for a study, does everyone here do empirical work? Is this relevant to everyone? Yeah. Okay, so you're determining your sample size. You can use a frequentist approach and do a power analysis, or you can use Bayesian approaches, which I'll explain in a second. And both of those are going to require thinking about this expected effect size or minimum effect size of interest. But you've got to consider what kind of size you're looking for in order to know how many people that you need. This third way of determining sample size is obviously really far from ideal, but for extremely expensive work like neuroimaging, or if you're doing mouse work, sometimes you've got this funding availability third option, which is obviously not the best scientifically, but pragmatically, that's sometimes what you have to do. So what effect size should I expect? Or what is the minimum effect size of scientific relevance? So the easiest way of doing this, depending on your field, is to just collect pilot data and learn about the data that it is that you're going to be pre-registering on anyway. Another way you can do this is to look at previous literature that uses a similar paradigm or a similar measure. It doesn't have to be exactly the same. If the measure is slightly different, you can just eyeball the bar chart, right, and say, well, the difference is this percent or this raw units, and then transform it to whatever it is that you're doing. None of this has to be perfect. You just want to be in a really good ballpark area. You can use logical reasoning. So, for example, I know based on some clinical standards that I need at least this big a treatment effect. Therefore, anything smaller than that is effectively zero to me. So you could do something like that. Or, as Zoltan's already mentioned, you can take the bottom of an 80% confidence interval from the literature. Depending on the domain that you're in, some of these will be relevant to you, some of these won't. But the purpose of putting them on the slide was just to highlight that there are lots and lots of tools available, and it's about thinking which one is going to work best for you. If you're doing a power analysis, most of those will work fine, but you do need to decide what that effect size you're working with is. And this, I think, there are lots of tutorials online and stuff how you determine your sample size using frequentist power analysis. Use something like G-power is nice and easy. But you can also take a different approach and just use raw effect sizes. So, for example, is everyone familiar with perceptual awareness scale? Yeah, it's like a 4-point scale. You ask people to rate the visibility of some stimulus. How well did you see this faint grating or something? And I can't remember what the actual labels are, but it's, had no impression at all, a very faint impression, blah, blah, blah. So let's say you're using a scale like that. Well, you could say my minimum interesting effect size is 1 point on this scale, one qualitative change in visibility, according to my measure, for example. And now you're not looking at Cohen's d or anything like that. You're not looking at standardized effect sizes. You're just looking in raw units at the kind of change that you reasonably think is meaningful for you. And if you want to do this kind of approach, then you can just run simulations to see how many people you need to get, say, 90% power. So you would say, for some range of sample size, say 12 to 150 per group, 10,000 times, you just sample data from normal distributions that differ on a pass scale of 1 point, run a t-test, and count the number of samples, simulations that give you a significant at p less than 0.05 test. So you can do something like this, and now you're not constrained to standardized effect sizes. This method is quite nice because it's theory-driven, so you're deciding what your bar graph or your line chart or whatever in the end is going to look like, and then basing your power analysis on that. You can also use Bayesian stopping rules. These are really nice. Are people familiar with Bayesian analyses here? It's like 50%. So here, you would set a minimum and a maximum sample size. So you say I have to collect, for example, at least 15 people per group. Otherwise, I'm just worried I'm measuring measurement noise. And say maximum 60 per group, because it's a really intensive study and realistically you can't do more than that. And now you determine a critical, that should really say Bayesian, so a critical Bayesian test for your hypothesis. So this is the main thing that you want to test in your study, your main prediction. So for example, it might be a Bayesian t-test between two groups. So it's like a t-test, but you're specifying a prior over your effect sizes. And you're trying to determine whether the evidence is more in favor of your H1 or your null hypothesis H0. And what you do is you start, once you get to 15 participants per group, your minimum, you just run your test, see if you've got enough evidence to stop collecting data, and if you don't, you keep going until you exceed some threshold, until your evidence is strong enough. So your Bayes factor, usually people do, is less than a third, so you've got evidence for H0, or greater than 3, so you've got evidence for your alternative hypothesis. This is completely different from frequentist. You can't do this with frequentist analyses, but you can do this with Bayes. So this is also a really nice approach. But for pre-registration, it doesn't hugely matter which one you choose as long as it's appropriate to what you're doing. It's just the process of making sure that you're planning all of these things in advance. So as an example of how you might determine the kind of effect size you need to look for, so this is a study that we ran where people had to discriminate whether there were more dots on the left or on the right. So that's what's in these boxes here. At the top, I don't know if you can see my mouse. No. So in the orange and blue boxes, we've got dots on the left, dots on the right. And the question is, which one has more dots? And then they give a confidence rating. And then participants complete 2 trials. And at the end, participants have to answer the question, in which trial, the first or the second, was your metacognition better? Were your confidence ratings more reliable? And so the prediction here is, if people are able to, if they know when their metacognition is good or poor, then metacognition will be better in the trials they reported as being the good ones. Okay, so what effect size should we power for in this study? So here we just based the reasoning on the measures we were using. So we have a measure of metacognition that we used. It's called M ratio, and it has a theoretical upper and lower bound of 0 and 1. Because it has, and I mean, this is Zoltan's approach for doing this. So you've got an upper and lower bound of 0 and 1. That means the biggest difference you could possibly have between the good metacognition trials and the bad metacognition trials is 1, right? Because in one group it's 1, and the other group, M ratio is 0. So that's the biggest possible effect. There's no way we're getting the biggest possible effect. That's ridiculous. So we just say we expect to have something between 0 and 50% of the maximum. So 0 and 0.5. Anywhere in that range is equally likely. I don't know. I really don't know what to expect. But then I set my Bayesian prior to be a uniform distribution from 0 to 0.5. So from nothing to 50% of the maximum. So it can be a little bit rough like this, and it shouldn't hugely matter if your effect is real, precisely what you decide, as long as it makes sense and the reasoning is solid. So you can base your effect sizes on your understanding of the measures you're using as well and how they should behave. So in the previous activities with Zoltan, did you have kind of predictions that you came up with as groups? Or were you more just talking around the issue? Did you come up with any? Yes? No? Kind of. Okay. Kind of. So for the prediction you would test, what kind of effect size do you think that you would come up with? Does anyone want to volunteer the topic they were talking about? Did anyone have a prediction? No? Okay, so I'll leave you to talk about it in small groups then. For the kind of remit you were talking about, what kind of a effect size would you expect to find? If this doesn't make sense because you didn't come up with a concrete enough question, say. Sorry? No, absolutely, you don't have the data. But for example, if it's in the clinical domain, you might say, well, in clinical, we have certain benchmarks or whatever. Or we have current best practice. And so we could base the effect size we're looking for on the comparison to current best practice. You could do something like that. Or if it's behavior, you could say, well, I'm interested in testing a question that's using a really established paradigm. So I should have lots of prior work I can look at and just base on that. Or I'm doing a completely new paradigm, developing a completely new paradigm, so I would have to pilot because I've really got nothing to work with. That level of discussion is fine. Okay, I'll leave you to talk about it in small groups.
Rather than having rather than us expecting with answers. Start to skip the process of being into
to force things, which is the force person when participants are
in essence, we're asking it's something far to this is some possibilities.
This is this part is there. Yeah. I'm going to check we could figure the difference.
What was the task actually?
yes. Then I will have to find very stuff. It's all good.
No one helped me much about establishing 5 minutes is being I'm so glad I didn't go through tests. I feel so better. Would you not? We didn't. Yeah, so also for the so the university. Oh, universities have a balance like this. Yeah, I know. It's impossible. I mean, now it's one thing now, but so what's the win plan?
Yeah, no.
Right.
All right, did anyone manage to come up with an approach for how you would... Did you manage to come up with an approach for how you would find effect sizes? Or does anyone have questions because it doesn't apply to what they were talking about? Any of the guidance? No? Yeah. How do you define it? Do you mean practically? So it's for you to do it. So it's just kind of a researcher choice. So you can, where is it? So you could use any of these approaches. It's for you to decide what it is that you're looking for when you design your study. So as you're writing, I mean, determining sample size you do with or without pre-registration anyway, but with the pre-registration, because you're committing to everything and you're explaining your reasoning, all of these decisions become really explicit. So depending on exactly what your topic is, you'll have a different approach to do it. But what did you have a domain in mind or a research question in mind? No? Is it a hard one? Is it? Do you want to share it? No? Okay, fine.
You could say
Then
getting a minimal effect of interest is... I'm sometimes surprised how ingenious authors have been in registry reports and coming up with something. It can be if there's a sort of a literature of relevant effects on the lower end, one of the lower estimates from that literature. Confidence interval, where you take the lower bound of the 80% confidence interval. But actually getting an expected effect size or a range up to a maximum is so much easier, which is one reason why I do base factors, because that's what's required for the base factor.
Oh, sorry, here we go. So once you've determined your sample size, and we can talk in detail at the end if you want about how to do that. In many cases, one of the first things you'll have to write in your pre-registration after sample size determination is your manipulation check. So you might have some manipulation or a paradigm that you're using or something, and you need to check that the very basics of what you were trying to do was effective. So how will you do that? How will you check that your paradigm worked? Or say you've got some attention manipulation, how do you know that you actually manipulated attention? So the example here is, say you're doing an ERP study. So you're predicting that some ERP component has a greater amplitude on easy than hard trials. A reasonable manipulation check here would be that on easy and hard trials, accuracy is higher on easy trials than hard trials, right? Because if people aren't doing better on easy things than hard things, then they're not really easy and hard. So this would be a straightforward manipulation check. If it doesn't come out, you just can't meaningfully test any sort of brain responses to difficulty in your study because you don't have a difficulty manipulation. It's kind of common. I see it a little bit less now, but there was one time when it was really popular that people would do some behavioral study and then do some neuroimaging or something with it. And the behavioral component would fail in the sense that there would be no behavioral effect of their paradigm at all. And they would still follow it up with neuroimaging. And they would say, oh, but in the brain, blah, 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 blah. But you don't really know you can't interpret that brain activity because you don't know what was going on in your study because your manipulation was not effective. So the manipulation check is really important. Does anyone do online studies, collect data online? Yeah, so it's always worth, because some of the online samples are so different to psychology students that people usually test, it's always worth also pre-registering checks on that sample. So is your sample behaving as you expect it to? It may or may not matter, but often it will. So when you're specifying your analysis plan, so now you want to test your key predictions. Sometimes people are testing something really straightforward, and all you need to say is, I'm going to run a t-test, and if this, then that. Otherwise, something else. But sometimes the project is much more involved, and the way in which you're going to test your question requires lots of steps, and you have maybe follow-up questions. So you can always pre-register conditional statements. That's absolutely fine. So if some analysis shows something, then I'll take this approach. But if it doesn't, or my data are distributed in this way, or I have this strange effect, then I'm going to take that approach instead. That's absolutely fine. It's always better to go for simpler options if you can, just because it's so easy to make mistakes when you have lots and lots and lots of steps. If you can't choose between two approaches, it can also be very useful to go for the one that's easier to specify in detail. So some analyses are just not that flexible, like ANOVA. You have your conditions and you have nothing else to do. And there are some other forms of analysis where you have to specify lots. So it's easier to go for the most simple and clear. An example of the problem with specifying things unclearly in your pre-registration. So this is one that I wrote with a PhD student. So I'm not going to go into everything, but we said that we would run a two-way mixed ANOVA on a measure of performance. And when we were talking about the different patterns of results that might come out, in the first case we said, so if the manipulation reduced bias, if the manipulation intended to reduce bias, made people worse, then we would find an interaction, a significant interaction, such that blah, blah, blah. In this second clause, which is one that we really cared about, we then said if the manipulation in the other group improves performance, then we should see that performance is higher as a result of this manipulation. So the way that we framed this was really problematic because we said that we needed this interaction term, so the second highlighted line here. We pre-register an ANOVA, we say that we're going to have an interaction, and then we list a bunch of t-tests that we actually care about. So this is mapping to something Zoltan said earlier. This interaction term in this ANOVA was completely redundant. We didn't need it. We didn't need to have a significant interaction at all. We actually didn't find one, and that was a problem because now we can't properly follow up with the t-tests that actually tests our prediction. So planning in advance exactly what it is you need to find, and then only putting that down, no extra tests, will help you avoid getting into a situation like this where you have to deviate from your pre-registration because you added something completely unnecessary. So as it happened, all these, the predictions came out fine, but not this interaction term that never mattered in the first place. So a lot of writing a good pre-registration just involves anticipating problems because when you think about, when you first think about what it is that you need to test, in some cases it's relatively straightforward what you need to do. What makes some people nervous about pre-registration is that when you get the data, there's going to be all these things you didn't think about that mean that your plan has to change. And there are certain ways that you can anticipate these problems in some cases and build them into your pre-registration. So again, not pre-registering more than you need is a really important one. If you're building on an existing study, so you're using a paradigm, for example, that's been used before, ask yourself how sure you are that it's going to replicate. And if you are very dependent on very specific patterns for your prediction, how confident are you that those patterns are going to come out again? If you're not, just pilot or something if that's feasible for you. And a third one is to consider any distributional assumptions you're making about your data. So, for example, if you pre-register that you're going to collect some variable, so a questionnaire measure or something, and you're going to mean center it so that the average is 0 before you put it into a regression. That's not going to be great if your variable is very heavily skewed. If it's distributed like this, mean is a terrible summary statistic for that data. So thinking about the distributional assumptions you're making and how sure you are about what your data will look like is another way to try and anticipate problems. And the best thing to do is to just, to the best of your ability, not pre-register anything that's going to not work. So the more robust analysis you choose, the less you actually need to worry about these problems. For pilot studies? Yeah, absolutely. Yeah, I mean, pilot studies are the best, and all you really need to do, ideally for a very strong pre-registration, is do a thorough pilot study. Not with, say, 3 participants, but say 15. Figure out all the different steps you need to take to process that data, to process and then analyze that data. Write it all in a code and then pre-register that. And you just basically repeat exactly what you did with your pilots. And then that's going to be a nice pre-registration, I think. Exclusion criteria are one of the biggest sources of researcher degrees of freedom. This is where it also gets quite hard. It's very, very easy to forget some component of your data or your participants, and then you have to, change your exclusion criteria after collecting the data because you can't retain some junky trials or something. So here, just maintaining a mental checklist of all the things you have to think about is probably going to be the easiest way. Or again, pilot data is always going to be helpful here. So you might need to exclude trials. For example, people who report really fast. If you collect timestamps, if you're presenting stimuli really quickly or something, you might exclude trials where the stimulus wasn't presented properly. Sometimes participants just aren't paying attention, right? So they give the same response every time. You're not going to want to keep them because they're not doing the task. The more experience you have with the kind of data you're working with, the easier this is going to be. But if you're doing a study you haven't done before, working in a domain you're not familiar with, then just asking colleagues about this is going to be really easy. Or downloading data from OSF and looking at the kind of weird things that you find there is going to make this much easier. So in a does anyone here do anything like visual perception or anything related to that? Yes? Yeah. Does anyone have like no familiarity with this kind of task, this kind of dot discrimination task? Is it okay? Okay, so if in a task where people have to compare the number of dots on the left and the right and say which one had more dots, and then you ask them to rate their confidence, what kind, and this was, I think this one was done online. What kind of exclusion criteria might you need? So I don't mean the actual numbers, like excluding people who responded faster than 100 milliseconds, but what kind of strange things might you find in this data? Sorry? Oh, yeah, absolutely. Yeah. Technical failure, disconnecting. Yeah. That's a nice one. That one's really hard. Yes. So in some cases, you would want to... So if you want to compare metacognition in two conditions, you might need to know that their metacognition is meaningful in the first place and then exclude, as you're saying. In other cases, it might be that you can't remove those participants because it's part of your actual question. So can people... It might be reasonable that people have very low metacognition on this task, and then you don't want to get rid of them because you're biasing your sample. But yeah, in some cases, absolutely. Any others? Yeah. Yeah, absolutely. So we did have some people just give the same answer over and over and over again. Yeah, that's quite common. We also had people who, they seemed to have kind of computer problems and the presentation of stimuli was taking a very long time. We had to exclude them. People who can't do the dot task at all, it was set up to be quite easy, so they should be above chance. So I'm not going to do this one because... No, okay. So again, in your groups again, have a think about what kind of data you might expect, and then accordingly, what you're going to need to be particularly careful about when excluding your data. Because this is, again, this is one of the more important things to put in your pre-registration.
So that would be useful feature.
Yes. 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 Is something to do with this house or like statements to do with her? Yeah. Yeah.
Right, so that there's enough time to do Q&A at the end. I'll stop you there. But so once you've got those sorted, this is going to be less relevant to some simpler paradigms, but if you're doing, for example, electrophysiology or neuroimaging or something like that, you might have a lot of data pre-processing. And this can also be sometimes a bit daunting to pre-register. So 2 general rules for pre-registering pre-processing that applies across the board. One is to just write codes that fully pre-processes your or analyzes your pilot data, because that way the analysis section of your pre-registration and the pre-processing section is just what you've done to your pilot data. And also, if your effect is real, it shouldn't really matter what you've done pre-processing wise, as long as you haven't done something completely ridiculous. You know, if your effect is critically dependent on some tiny parameter in a chain of 10 pre-processing steps, it's probably not real. So, for example, if it's reasonable to just go with the default options if you're doing fMRI and SPM, wherever default is reasonable, it's just an easy way of setting that pre-processing so that you don't have to worry about it too much. There'll obviously be cases where you can't do that, and it is really important, and this doesn't apply there. But in general, you don't need to plan every pre-processing step in. You don't have to think them all through if the defaults are going to be fine. Sometimes you don't actually need to do a pre-processing step. For example, if you're doing fMRI, if you don't need to do slice time correction, just don't do it, because the less you do to your data, the better in many cases. And you just reduce the number of ways in which you can make a mistake. But otherwise, again, if you can use some robust method, that's going to make your life easier later, because there's less to think about when you're planning your analyses. So for behavioral data, I've just written, this is going to be incomplete, but a checklist for the kind of pre-processing steps you might need to consider and therefore write in your pre-registration. So how are you going to treat missing data? So that might be if somebody hasn't made a response, or it might be a technical failure. It might be that you're comparing different kinds of responses in different conditions, and you might have empty cells, so somebody's just never done some combination of things. What are you going to do with those? How will you treat skipped trials or skipped questionnaire items? Are you going to fill them in with the mean or the median, or are you just going to ignore them? How will you calculate your dependent variables with equations? So sometimes there are multiple ways to estimate a measure. You need to say which one you're going to use. How will you evaluate the results of attention checks? So in some cases, particularly online studies, you might present some trivially easy question. And if people can't answer it, then you exclude them. maybe you do this 3 times. How many times do they have to fail to get kicked out of your data set? Will you need to transform your data? Will you need to screen for outliers or problematic responses, like implausible ones? Do you need to screen for technical failures? And if so, exactly what would you do? So this all goes in your pre-registration as well. And so as an example of what happens when you don't do this properly, this is also one of mine. So suppose you're collecting data from some clinical subscale, like a questionnaire. And suppose you're collecting this data online, which it is. You need to think about whether the distributions you see in the literature on either patient groups or really large samples in the lab are realistically going to reproduce when you collect the data online. So this is data I collected online. So I was interested in this dissociative experiences scale questionnaire, and there were these subscales, one on depersonalization and one on amnesia. And what I had looked up before writing the pre-registration was the distribution of the overall questions, which is kind of okay, right? This is this DES. That's kind of okay, and I pre-registered I would do a median split, compare highs and lows, and do linear regression. But the data I got with these disgusting skewed distributions, and medium split is just 0 and everything else, and it was completely implausible to do any of this, right? I just had to throw the whole pre-registration in the bin. So this is a case where piloting would have been really expensive because online studies are really expensive. But thinking really carefully about whether it makes sense that your subscales are really going to have the same distribution as your as the overall questionnaire is a way of getting out of this problem. Another one is to just not pre-register tests like median split and do something more appropriate. A lot of what both, or almost everything that Zoltan and I have already covered applies just as much to neuroimaging, physiology, and modeling. There's just because whenever there are more steps to an analysis, there's just more stuff you have to think about in the pre-registration. But all the logic is the same. So you have many, many more steps to specify, and you have many more and accordingly, for the same reason, you have many more research degrees of freedom. But otherwise, it's all exactly the same process. So if you are, sorry, there's some sort of bug. So if you're going to do something like fMRI or EEG or modeling or any kind of analysis or bootstrapping with behavioral data, any kind of analysis where you are using random number generators, just make sure you pre-register your seed so that everything can be reproduced. Make sure you pre-register your software versions because sometimes algorithms change. But otherwise, it's all the same process. Because it's so involved and there are so many steps, it's really helpful to have a collaborator that is familiar with that paradigm or method or technique if you're not. So if it's, say, you want to pre-register some fMRI study and you've never done fMRI, bring somebody in who knows what fMRI data looks like. And this might not be the head of the lab who doesn't handle the data anymore. This might be like the postdoc who has done all of the data analysis for the last 5 papers, right? Because you need somebody who knows what happens when you actually have this data in front of you before it's worked with. And when there are lots of steps to your pre-registration, just getting feedback from colleagues is always going to be really helpful as well, because they'll check whether you've missed something. Registered reports mostly fixes this problem because your pre-registration plus some other bits go under peer review. But if you're pre-registering without that, then getting informal feedback from your peers is going to be really useful. So again, this was in the previous section. It applies here as well. If you are pre-registering something like, say, EEG, just analyze your pilot data and write up what you've put in your code. And it's going to be the most thorough pre-registration you can have. And the specific choices you make, again, shouldn't matter hugely, as long as they're reasonable choices.
In general,
Mm-hmm. Yeah. Mm. Mm-hmm. Yeah. Yes. Absolutely, I completely agree. So maybe I wasn't clear. I didn't actually mean upload the code. I meant translate the code into words and write it. So I will use this algorithm with these parameters. Blah, blah, blah. But yeah, code review is a really big problem. And then you've got kind of orthogonally to pre-registration and all of this, you've got this computational reproducibility problem, which is very severe. Yeah, I agree with you. So for EEG, a kind of, again, rough checklist. I'm sure I missed things, and do let me know if I've missed anything. You would need to fully describe the pre-processing steps you're taking, including the parameters. Where you can, try to make it computationally reducible, so somebody else can repeat what you've done and get the same answer. So one way, something that sometimes comes up with EEG is you need to do artifact rejection. So links, muscle movements, things like that, cause deflections in the EEG, noise. I like removing them manually, eyeballing it. There's this downside to that, which is nobody else can reproduce what you've done. So other people use ICA or some algorithm. Sometimes it's useful to not do things manually so that it's computationally reproducible. I think sometimes if one technique is just clearly better than another, you're not going to sacrifice a good technique just for a neat pre-registration. You would also need to fully specify all your outcome variables in detail. So for example, I want to, yeah?
I'm sorry, I missed. Place where you need to do something.
So personally, I would just say I'm going to do it manually. On the, and these are going to be my criteria. So for example, I'm only interested in this period of the epoch. So I'm only going to exclude on the basis of what's going on in that epoch, plus or minus a tiny bit. And if there's noise...
Could I interject?
Way later. Yeah, you might disagree with what I'm saying here.
Yeah, when I get a registered report where the person says they're going to manually do something, so with EEG, I see analytic flexibility. Which in other words, you could change things in the light of the p-values or base factors that you get, and take that garden of forking paths with the multiverse towards the results that you want. So this is what we're trying to tie down. So you can either come up with... So one rule is you want to... And this is sort of more strictly enforced for a registered report, because with the pre-registration, you just, as you say, just put down what you want. But let's talk about ideally what you put down in pre-registration. So one rule is you want to make it so that anyone else would come to the same results as you did, given the link to the open raw data and the specification that you've given. Now, that might not always be possible. So remember I talked about blinded analysis. So what I have had with an EEG paper I was dealing with, they said they'll do a blinded analysis, and they have to specify how the blinding is done. So you just want to make sure that the choices that are made are not made in the light of knowledge of the results that they produce. So as long as you can ensure that that happens, that's fine. You're still doing bias control. But you just have to specify in the pre-registration how it is that you're going to ensure that that happens.
So did they have somebody else do the manual artifact?
Yes.
Because it's blind at the point at which you're doing it.
As long as you can specify that.
So burdensome. I can't imagine somebody going back and p-hacking manual artifact rejection because it's such a torture over like 64 channels.
Yeah.
With 200 trials.
Yes, in that particular case.
You really have to be motivated.
Yeah.
But so you would have somebody else do it blind to the conditions and then pass that data on. That makes sense.
Cool. But you make it. So that's for a registered report. We try and do it strictly.
So say you're analyzing ERPs, you would need to pre-register. How are you going to calculate the baseline? This also applies to oscillatory stuff too. How are you going to calculate your baseline? If you're doing ERPs, what's the time window you're looking in? So you might be interested in some component, say N170. Don't bank too hard that it's going to come really cleanly in that at that time point, because latency, because it probably won't. So you need to think about how you're going to deal with that. And you know, if you're correcting over areas, how are you going to do that? And if you're collecting data over multiple runs or sessions, so say you stop collecting data, participants have a break, when are you going to combine it all? Are you going to pre-process it together? Are you going to pre-process it separately? These are quite hard to think of a priori sometimes, but again, if you just analyze your pilot data, these are all things you're going to do automatically, because you can't get to the point at which you're testing your hypothesis without answering all of these questions. So sometimes it's hard to think about it in abstract, but when you actually mechanically go through it, you start to notice all of the things that you've done, and that's why working with pilot data is so useful. There's a problem on this slide. So just as like little checklists, physiology I've put together with neuroimaging, because most of the kind of decisions are going to be very similar. So in your pre-registration, you're noting all the pre-processing steps that you're taking, the parameters for any algorithms you're using, like for fMRI, smoothing kernel, any encoding, so how you're labeling conditions and stuff, things like that, how you're labeling any categorical data. Random seed, software, measures, regions of interest, labeling of regions of interest for MRI, so which atlas are you going to use? Re-referencing data, baselines, all of this has to go in. For modeling, similar, fitting procedures, how you're going to fit your models to your data. What are your key measures? What are your model evaluation metrics? So how are we doing for time? So an example of how you might structure a pre-registration for a more complex analysis. So in this paper, we wanted to test a theory of subjective time, which is subjective duration is just the number of salient events in perceptual classification. So how much stuff is going on in the perceptual system. And that amount of stuff is the amount of time, broadly. So we had participants in the scanner rating the duration of some videos. And if the theory is correct, then their bias in how they rated this duration, like was the video, did it feel long or short, should be predicted by the number of salient events, so things that happened, in visual cortex bold, but not auditory or somatosensory. Those were the control regions. So we did a pre-registration for this, which took quite a long time. So we say what we're analysing our data in, so SPM12. It shouldn't really matter hugely how you're pre-processing this data, so we just said default settings for all pre-processing unless otherwise specified. And then where we deviated, I've just put it in white. But you've just got nice, simple bullet points that outline your pipeline for how you're pre-processing your data. You don't need to explain anything. You don't need to go into detail about any of it. Just nice and easy, so brief, so that whoever reads it can check that you followed it. One part of our analysis that wasn't the critical part involved just doing like a GLM and ANOVA type analysis on this data. So we explain how we're going to do it. Again, relatively briefly, all the decisions that have to be made when you create these models in SPM, we explain how to do it. We explain exactly what we're modeling, so all the variables that are going to be going in, and how they're going to be modeled. So that's this AR1. Then there's the second stage of running these models in SPM. So we explain how we're going to correct over all of these voxels in the brain. We explain how we're going to label any significant ROIs. So which atlas are we going to use? How are we coding our contrasts? So we want to do this ANOVA. We have a couple of things we want to test. We're going to test them by running the contrasts in the yellow box. And then for modeling, we explain how we're going to model the data, what parameters we're going to use based on pilot data, and kind of step by step how we do it. It looks a lot like a method section. It looks basically almost exactly like a method section, which is what you're aiming for. Plan to allocate the time that you'd spend analyzing your data after collecting it to writing the pre-registration, because analyzing the data is going to be really, really easy once you've done the pre-registration. You've done all of the hard work. So writing the pre-registration for the neuroimaging took about 6 months. And I think analyzing fMRI data and doing some modeling is about 6 months worth of work, approximately. Sounds about right. So you're just doing that piece of work before collecting the data instead of afterwards, that's all. You save that time. Be prepared for much, much smaller effect sizes than you're used to when you pre-register more complex things. So for that one, we had a correlation between two critical measures, so amount of time and amount of stuff. And it was significant and everything came out as we planned and everything, but the Pearson's R was 0.03, which is really, really small. And the parameters we chose for our model were poor, so we did a sensitivity analysis where we ran it over all sorts of different parameter choices, and we were in the wrong space. We were somewhere really suboptimal. That's all fine. It's all things that you just prepare for when you do the pre-registration. It's just you can be really confident that you haven't hacked your way around to get the answer. And then the answer, your effect size will be much less exciting than you're used to. That's okay. Make sure you get feedback wherever possible. Consider registered reports. It makes things much easier, not doing it yourself. So a registered report is a little bit like a peer-reviewed pre-registration. So you write the whole paper from introduction to what you're going to do in your results without having the data. It goes under peer review. And then once it passes through peer review, you get stage 1 conditional acceptance. So you can then go collect your data, you analyze it according to what you said you would do in your registered report. You interpret your results in line with what you said you would do initially. And now you have your complete paper. So it's pre-registered in a much more rigorous way. But the important point is that some journals, when you do this process, you get acceptance at stage 1 before you get the data. So even if your results are messy or horrible or null or whatever, things journals usually don't like, they commit to publish anyway, as long as you follow. I'm sure there's stuff you want to add there sometime.
No, that's good.
Is that okay? Okay.
So I guess it helps get rid of publication bias. If the design was a good design to address a scientifically relevant problem, then the journal saying we'll go with that no matter what the results.
It's a solution to the problem that nobody checks your pre-registration, as you mentioned at the beginning, and that pre-registrations can be problematic. If you're pre-registering modelling, you'll usually need some pilot data or an established result to build on, simply because there's just no point in modelling nothing. So you're probably going to want to model something, and then whatever is motivating your project that you want to pre-register, you can draw on that to write your pre-registration. if you're fitting a model to data, for example, drift diffusion models or something like that, something to think about when you write your pre-registration is you're probably going to want to check that your model fits are satisfactory, like before you compare parameters across conditions or something. So you need to plan how you're going to do that. And then where relevant, so probably not really for drift diffusion modeling, but if you are uncomfortable committing to some set of parameter choices for your modeling, you can always say that you'll perform some exploratory parameter sweep. So you then rerun your analysis over all the parameters and just see whether your conclusions were based on being in some very weird parameter space or whether it's robust to your choice. This is a repetition of the checklist I had before, but they're all just merged here. We'll share these, so if it's helpful to have these checklists, we'll share them with you. So that's the end of the talk part. I don't know how we are for time. We don't have very much. Does anyone have questions for Zoltan or me? Or specific examples of things you want to pre-register? Do you know?
Some funders, I haven't heard of funding for
ASSE
pre-registration, but there is for registered report. So some funders do deals. Templeton does a lot of this in consciousness science. But they just had a sort of a splurge on it, and they Templeton continues to do it with adversarial collaborations with the different theories of consciousness. Other funders have done it in other disciplines where the best way of doing this is and this was a Templeton initiative with ASSC about a year or so ago. It's now finished. We're getting your stage 1 registered report approved after initial screen is the process of getting the funding from Templeton for doing that study. Because it's ideally combined in one step so that the funding decision and the acceptance of the stage 1 protocol is the same decision. So I'm not aware of a sort of a standard open-ended process of doing this. It comes up as special initiatives by different funders now and again, but it's ideally best because otherwise you get some protocol or procedure approved at the funding stage and then the registered report stage it gets changed when you put it through the stage 1 through the second lot of reviewers. Of course, it can be done that way, but it's much better to combine the process and have it explicitly funded into the workload you put into the grant, because the registered report is more work, and you're going to get out less papers this way. I consider that worth it. See, that's what I do, but it's good to have that built into the funding model so that it's understood that this is the level of output you're going to create.
Was to be accepted as the stage 1 registered report in one of the 5 or 4 journals who were like part of this.
Yeah, Templeton have been really good at this in different initiatives. They're doing.
You have to like consider the period of like writing and going through all this like rounds of reviews and rewriting the proposal. But then after you have the data, it's not like it's quite faster than making it. So it's like more and then you get the faster. Well, I mean, in our case it was 2 years, but it's because we had to wait because we didn't have enough participants. So with like enough participants, it would be faster. It would be.
I currently have a Templeton grant which involves delivering a registered report. But that was part of the deal from Templeton. But I'd also do it in writing other grants. I'd say this is going to be a registered report. It's going to take this amount of time to do, and I'll build that into the grant. Even if it's not an explicit initiative on the part of the funder, you can still put it in as part of your funding application.
Any other questions? Okay.
labs where students are the rules of the ground rules.
Yeah.
There's so many questions you can ask with one data set.
Mm-hmm.
And that becomes sort of hard to figure out whose question
Yeah.
it is. And like
Sorry.
the main hypothesis of the project
Mhm.
we registered and having it done and having
Yeah.
like a confirmation, but afterwards, why is it leaking? Like, because you really want to press that limit as much as you can. You know
Yeah.
what's the what you can extract out of that data, but you've had like one pretty study publication, but what can you do in that stuff?
I mean, you can always pre-register in the same way that you would if the data hadn't been collected yet. The problem is that you have access to the data already. One solution to that is that the PI handles the data themselves and doesn't share it with lab members until they've done their pre-registration, and then gives access. So you have, for example, a pilot data set, you cut out 10% of the samples. You can play with that, and then the remaining you only get access to if you've done a pre-registration. But then you need the PI on board for that, or whoever it is that owns the data. Otherwise, you can still pre-register. It's just, I mean, in PCI, it's just called level of bias control.
Yeah.
To what extent are you able to protect yourself from the bias you're exposed to? Sometimes you can't. That's okay. You can still write the pre-registration.
Yeah, we regard the classic case, full bias controls, where the data doesn't exist yet when you do the pre-registration. But to enable other sorts of situations and flexibility, we allow people to pre-register when there does exist data, but we give it a lower level of bias control. So there could be data you don't have access to yet because you're not allowed access until you apply for it, or it hasn't been released yet. That gives you some bias, sort of external sense of bias control. Or it could be a data set that you have and you could have access, but you're just swearing that you haven't. We allow that, but we say that's the lowest level of bias control. But still, you can get some of the benefits of pre-registration from it. You just have to be clear in the pre-registration, what level of bias control we're talking about.
Question. So, when one study also implementation when you have collaboration, so you have multiple paradigms on the same population, but that actual question, but it's on the same population. What would be the best practice to register results? That would be okay?
Yeah, you can do 3 separate pre-registrations. If they're dealing with different issues, I'll probably do different pre-registrations.
Okay. My question is, so we're going to really compare novice with control to, say, experienced practitioner. Would it be okay to 40-40 in this group? And we use, of course, a couple of control ingredients in here to make just to get also a sample size, but because our discussion is on the contrast in the population, is it okay to estimate effect size based on the first control and then to register? That would be okay when you do as a practice?
Yeah, in fact, I'd often do that. So I might run a pilot, which isn't the full design, but there might be, let's say, I'm expecting the practitioners to be less on some variable. Then I could run the control, and I could say, let's say it's depression or something, the average level of depression. I'll say, whatever effect it's going to be, it's going to be less, the depression is going to be less than these control subjects. That helps me define the prize of my base factors. I don't need to run the full design to get the prize out. So no, that's a really useful way of doing things. Run the pilot, that helps you fine-tune all your models, including your how you're modelling effect size for power analyses, base factors, for instance.
Can you remind us how you get the PowerPoint? Are you going to share them?
Yeah, we weren't allowed their emails, were we?
No.
But you can email us. If you email us, we'll email you. Yes.
No, I asked if we could get all of your emails so that find out what kind of thing everyone does before we did the tutorial, but it's not allowed. So we can't email you. Oh, yeah. Can't remember yours, Zoltan. Are you just Z?
DNS or z.dns.
I don't know if ASSC share all of this stuff. They usually share it afterwards on the website.
Emails.
No. Presentations.
PowerPoints.
Yeah.
You know, I
Some
don't think they
conferences
normally do.
Okay.
Don't think they normally do. Okay. But if you email us, you'll get it.
You can send us the slides?
Yeah.
Yeah.
Thank you.
I was also planning to YouTube it. Are you all happy that we YouTube it? People, your voices will probably be dim, but they might hear some of your voices. So just want to check. No one will know who you are, presumably. Is that all right?
Yep.
Yeah. Right.
Right. Thank you.